Welcome, my dear students. Today we are stepping into one of the coolest navigation systems in cell biology. In the first part of protein sorting explained how cells use their GPS system. We will explore how every newly made protein in your body knows exactly where to go. Thanks to tiny built-in address labels, it's like the cell's own courier service working with perfect accuracy. Let's dive in and see how this incredible system keeps life running smoothly. Introduction. Most proteins basically are made by ribosomes within cytoplasm, but some proteins are made inside mitochondria and chloroplast themselves. But proteins made in the cytoplasm need to be sent to the correct place in the cell to work properly. To reach their destination, proteins from the cytoplasm or cytosol must cross membranes of organelles like the nucleus, or mitochondria. Each newly made protein so carries a specific address level or sorting signal. This sorting signal is a part of the protein's amino acid sequence. This signal tells the cell where the protein should go, for example, nucleus, mitochondria, etc. The process of recognizing and sending proteins to the right compartment is called protein sorting. Dr. Günter Blobel discovered that proteins contain built-in signals that guide their transport and localization in the cell. This idea is known as signal hypothesis. So what is signal hypothesis? Signal hypothesis means the protein contain a inbuilt or built-in signal that guide that protein to their transport and localization inside the cell. And this concept was developed by Günter Blobel, David Sabatini and Bernhard Doberstein in 1971. According to this hypothesis, the signal sequence helps the protein move to specific organelles through the correct pathway. Examples of destinations guided by sorting signals include the nucleus, endoplasmic reticulum, mitochondria, plastids, and peroxisomes. From the endoplasmic reticulum, proteins can be further transported to other parts of the cell such as the Golgi apparatus or plasma membrane. Three basic modes of transport. Sorting signals, which are also called as signal sequences, are short stretches of amino acids we know. They act like address labels showing where each protein should go inside the cell. Different cell compartments like nucleus, endoplasmic reticulum, or mitochondria have different address levels. Sorting signals control how proteins move from one compartment to another, and there are three main types of protein transport inside the cells. First one is the gated transport, second one is the transmembrane transport, and third one is the vesicular transport. This is the image showing the transport through nuclear pore. This is transport across membrane, and this is transport by vesicles. The transport through nuclear pore is the example of gated transport. So gated transport happens between the cytosol and the nucleus. Proteins move through tiny openings called nuclear pore complexes in the nuclear envelope. These pores act like selective gates. They allow small molecules to pass freely but control the movement of larger ones. But large proteins need a special nuclear signal for transport. This is the image showing different modes of protein sorting. Now, transmembrane transport, proteins move across a membrane into organelles like endoplasmic reticulum, mitochondria, peroxisomes, or plastids. They are under transmembrane transport. Specific transport channels called translocators help move the protein across the membrane. The protein usually has to unfold to pass through the translocator. Vesicular transport, proteins are packed into small membrane covered sacs called vesicles. These are the vesicles. Vesicles carry proteins between compartments that are connected by membranes such as Golgi apparatus, endosomes, lysosomes, and cell surface. Each vesicle knows where to deliver its protein based on recognition of sorting signals. In every type of transport, that is gated transport, transport membrane transport and vesicular transport. Shorting signals on the protein are recognized by matching receptor proteins. These receptors ensure each protein reaches its correct destination in the cell. Signal sequences and their role. Signal sequences guide proteins to their correct location inside the cell. They are short stretches of amino acids, usually 15 to 60 amino acids long. Most signal sequences are located at the beginning, that is N terminus of the protein. Once the protein reaches its destination, the signal sequence is often cut off by special enzymes called signal peptidases. Some signal sequences are found inside the protein instead of at one end. These internal sequences stay as part of the protein. But in some proteins, several internal sequences combine to form a 3D shape on the surface called a signal patch, which helps direct the protein. Every signal sequence acts like an address tag that tells the cell where the protein should go. Examples of signal sequence: proteins going to the endoplasmic reticulum or ER usually have N-terminal signal that contains 5 to 10 hydrophobic amino acids. Some proteins move from the ER to the Golgi apparatus, but if they have a special 4 amino acid signal at their C terminus, they stay in or return to the endoplasmic reticulum. Proteins going to mitochondria have signals with alternating positively charged and hydrophobic amino acids. Proteins going to peroxisomes usually have a signal mode of 3 specific amino acids at their C terminus. These are the examples of different signal sequences, which signifies import into nucleus, export from nucleus, import into mitochondria, import into plastid, import into peroxisomes, import into ER and return to ER. Gated transport: protein import into the nucleus. What is the mechanism? Signal for entry. Proteins that need to enter the nucleus have a nuclear localization signal or NLS. The NLS is a short stage of positively charged amino acid like lysine and arginine. The signals acts as a label directing the protein to the nucleus. Nuclear envelope and NPC. The nucleus is enclosed by the nuclear envelope, we know, which contains nuclear pore complexes or NPCs, and these NPCs control what enters and exits the nucleus. Import receptors or importin. Proteins with NLS are recognized by nuclear import receptors, also called importins. Each import receptor is specialized for certain cargo proteins with specific NLS sequences, and these import receptors bind the NLS on the cargo protein and FG repeat sequences on NPC proteins. FG repeat proteins, NPC or nuclear pore complexes, contain FG repeat, that is F indicates phenylalanine and G indicates glycine repeat. They form a pathway through which import receptors and their cargo travel. The receptor cargo complex then moves by repeatedly binding and releasing FG repeats. Transport into the nucleus. Once inside the nucleus, the import receptor releases the cargo protein. The receptor then returns to the cytosol to transport more protein. Sometimes adapter proteins help link the receptor to the cargo. Energy and directionality. The role of RAN GTPase. The transport process requires energy from GTP hydrolysis. The key energy regulator is the RAN GTPase. We know this is an enzyme which acts as a molecular switch, and this RAN exists in two forms: RAN GTP mainly in the nucleus and RAN GDP that is mainly found in the cytosol. RAN regulatory proteins. RAN GAP, that is GTPase activating protein, is found in the cytosol and it can convert RAN GTP to RAN GDP. Then RAN GEF or guanine nucleotide exchange factor is found in the nucleus and converts RAN GDP to RAN GTP. RAN gradient and direction of transport. The separation of RAN GAP and RAN GEF creates a RAN GTP gradient across the nuclear envelope. This gradient ensures directionality. In the cytosol, RAN GDP site, receptors can bind to cargo. In the nucleus, RAN GTP site, RAN GTP binds to receptors causing them to release cargo. Thus, cargo unloading always happens inside the nucleus. So the summary of the steps: proteins with NLS binds to import receptor in cytosol, and receptor cargo complex then moves through NPC via FG repeats. Inside nucleus, RAN-GTP binds receptor to cargo released, then receptor returns to the cytosol. RAN-GTP is hydrolyzed to RAN-GDP, and directionality is maintained by RAN-GTP distribution. Nuclear export, that is transport out of the nucleus. Purpose of the nuclear export: large molecules such as ribosomal subunits and various RNA molecules like mRNA, tRNA, rRNA need to move from the nucleus to the cytosol. This movement occurs through nuclear pore complexes using a selective transport system. Nuclear export signal, or NES: molecules that must leave the nucleus contains a nuclear export signal, or NES, and the NES then acts as a tag that directs the molecule to the export pathway. Export receptors, or exportin: proteins with NES are recognized by nuclear export receptors, also called exportin, and exportins bind to two things: the NES on the cargo molecule and the FG repeat proteins that line the NPC, which help guide the cargo through the nuclear pore. Family relationship with the importin group of protein. Exportin are structurally related to importin, the nuclear import receptors, but both importin and exportin belong to a common gene family called as karyoferrin. Karyo means nucleus and ferrin means which carrier protein. In each there are about 14 karyoferrin genes, while in animal cells the number is much higher. Functional similarity and opposite direction. Nuclear import and export work in similar way but in opposite directions. Import receptors on importin bind cargo in the cytosol, release it in the nucleus and return to the cytosol. Then export receptors or exportin bind cargo inside the nucleus, carry it out through the NPC and release it in the cytosol. Dependence on RAN-GTPase. Like import, nuclear export depends on RAN-GTPase for directionality and energy. Inside the nucleus, RAN-GTP binds to export receptors and promotes cargo binding. Once in the cytosol, RAN-GAP triggers GTP hydrolysis, converting RAN-GTP to RAN-GDP, and this change causes cargo release on the cytosolic side, completing the export processes. Recycling of components. After releasing the cargo, the export receptor and RAN-GDP return to the nucleus to be reused. Inside the nucleus, RAN-GEP regenerates RAN-GTP to maintain the transport cycle. During mitosis, the nuclear envelope disassemble and temporarily halting nuclear transport process. And the nuclear proteins RNAs mix with the cytosol until the nuclear envelope reforms after cell division. Transmembrane transport. Protein imported into mitochondria. Source of mitochondrial protein. Most mitochondrial proteins are encoded by nuclear DNA and synthesized in the cytosol. After being made, they are imported into mitochondria post-translationally after protein synthesis is complete. Precursor proteins and signal sequences. Newly synthesized mitochondrial proteins are called mitochondrial precursor proteins. These proteins carry specific signal sequences that direct them to the correct mitochondrial compartment. Depending on the protein, the signal sequence can be at the N terminus and removed after import, or internal and retained permanently. Types of signal sequences. The signal sequence for proteins entering the matrix space forms an amphiphilic alpha helix. In this helix, one side has positively charged residues and the opposite side has hydrophobic residues. This structural pattern, not the exact amino acid sequence, is recognized by mitochondrial receptor proteins. Mitochondrial subcompartment. Mitochondria have four main regions, that is outer membrane, inner membrane, intermembrane space and matrix space. Specific signal sequence direct proteins precisely to one of these compartments. Protein translocator complexes, mitochondrial protein import depends on multi-subunit translocator complexes located in the membrane such as TOM complex, that is translocase of the outer membrane, Team 23 and Team 22 complexes, translocase of the inner membrane, SAM complex, sorting and assembly machinery, or OXA complex, oxidase assembly machinery. Functions of major complexes, TOM complex, that is present in the outer mitochondrial membrane and it is required for the import of all nucleus encoded mitochondrial proteins. Passes proteins into the intermembrane space and helps insert some into the outer membrane. This is the Tom complex and SAM complex. Now we must learn about the SAM complex, Team 23 complex, Team 22 complex, and OXA complex. So SAM complex is it works with TOM for beta barrel protein, which are abundant in the outer membrane, and it helps fold and assemble these proteins properly. Team 23 complex transport soluble proteins into the matrix space, and it also helps embedded transmembrane proteins into the inner membrane. Team 22 complex, it inserts a specific subgroup of inner membrane proteins such as transporters for ADP, ATP and phosphate. OXA complex, it inserts protein synthesized inside mitochondria into the inner membrane and also assists some imported protein that first enter the matrix. Energy requirement and directionality. It import into mitochondria requires ATP and membrane potential across the inner membrane. This energy drives the transport and proper folding of proteins once inside. So what is the key concept? Signal sequences are both necessary and they are sufficient for proper targeting. If attached to an unrelated cytosolic protein, they can still direct it to the right mitochondrial compartment. So my dear students, that brings us to the end of the first part of protein sorting chapter, how cells use their GPS system. You have seen how proteins carry their own address levels and how the cell's transport machinery reads these signals with amazing precision. This tiny molecular GPS keeps every compartment working perfectly. Thank you so much for watching and learning with me. Stay curious, keep exploring, and I will see you in the next part.